Myself, Dr. P. Shokumar, working as Associate Professor in Biotechnology in Dr. M. S. Swaminathan Agriculture College and Research Institute. It is one of the constituent college of Tamil Nadu Agriculture University. Today, I am going to talk about the transcription and post-transcription processing. Now we can see the transcription and translation in both prokaryotic and eukaryotic cells. In a prokaryotic cells, the transcription and the translations are coupled or it may be occur in one place. That is, the translation begin, the mRNA is still being synthesized. In a eukaryotic cell, the transcription occur in the nucleus and the translation occur in the cytoplasm. So the below picture show how the transcription and the translation occur in the prokaryotic cells. Coming to the eukaryotic cells, the transcription translation are spatially, temporarily separated in the eukaryotic cells. That is, transcription occur in the nucleus to produce the pre-mRNA molecule. The pre-mRNA molecule typically processed to produce the mature RNA. In case of prokaryotes, there is no need of the processing of the pre-mRNA to the mature RNA. So which is exit the nucleus and is translated in the cytoplasm. So the below picture show how the transcription occur at a nucleus and the translation occur at a cytoplasm. In these slides, we can see the different genes for different RNAs. There are 4 types of RNA, each encoded by its own type of gene. In any cell, only some of the genes are expressed in the particular stage or particular time, and some of the genes are not expressed or maybe in the off condition. So we can see the one by one the RNA types. First one is the mRNA. mRNA or otherwise called as messenger RNA. It encodes amino acid sequence of a polypeptide. The second one is called as tRNA, that is the transfer RNA. It binds the amino acids to the ribosome during translation at cytoplasm. Third one is the rRNA, that is a ribosomal RNA. It will bind the ribosome protein. It makes up the ribosome, the organelles that translate the mRNA molecule. snRNA is nothing but or otherwise called as small nuclear RNA bind with protein. It form the complex that are used in RNA processing in eukaryotes. It is not present in the prokaryote cells. Coming to the transcription process, the RNA synthesis. Transcription is nothing but DNA is converted to form a RNA molecule. The process called as the transcription. Here, the RNA synthesis involves separation of the DNA strand and synthesis of an RNA molecule in the 5' to 3' direction by the enzyme is called RNA polymerase. So RNA polymerase is normally involved to synthesize the RNA molecule in the direction of 5' to 3. It is using one of the DNA strand as a template. So the parallel DNA acts as a template for synthesis of RNA molecule. In a complementary base pairing, the A, adenine, thymine, guanine, and cytosine on the template DNA strand specify the U, A, C, and G respectively on the RNA strand being synthesized. The following picture shows how the transcription occur in 5' to 3' direction, how the RNA polymerase bind with template DNA to synthesize the RNA molecule or maybe the transcription process. This slide shows the complete transcription of an RNA molecule. So transcription normally start at a promoter site. The promoter is normally present in the proximal region of the molecule, DNA molecule. So the transcription begin at the promoter. It proceeds through the coding regions and ends at the terminator. So terminator is normally present in the distal end of the DNA molecule or genome. So the following picture shows how the transcriptions occur by the binding of the RNA polymerase. Coming to the mRNA in the prokaryotes. Now we can see the mRNA prokaryotes and mRNA in the eukaryotes. Now we can see the mRNA in the prokaryotes. The sequence of the prokaryotic protein coding gene is collinear with the translated mRNA. That is, the transcript of the gene is the molecule that is translated into the polypeptide. Here you can see the picture. The top one is called, left side is the DNA molecule. The left side is the proximal end having the promoter, and the right side having the terminator. In between the promoter and terminator, we have the RNA coding sequences. The RNA coding sequences go for the transcription, that is the coding, protein coding sequences. Then the transcription translated to form a peptide that is called as polypeptide. Coming to the mRNA in the eukaryotes, the sequence of the eukaryotic protein coding gene is typically not collinear, but in prokaryote, this is a collinear fashion. Then the translated mRNA, that is the transcript of the gene, is a molecule that must possess or processed to remove the extra sequence, that is called a syndrome. So in mRNA, here during the processing, the introns are removed or intervening sequence are removed in the eukaryotic cells before it is translated to form a polypeptide. So the picture shows how the RNA coding sequences transcript and to form a translation and finally to form the polypeptide. After getting the pre-mRNA, we go for mRNA processing. So we can do some processing mechanism, adapt the mechanism, then we can get the protein coding sequences. The protein coding sequences are translated to form a polypeptide in case of eukaryotes. Coming to the RNA polymerase, this is one of the important enzymes involved in the transcription process. The enzyme responsible for the RNA synthesis is DNA-dependent RNA polymerase. The prokaryotic RNA polymerase is a multiple subunit protein of nearly 480 kilodalton bases. In eukaryotic system, have three kinds of RNA polymerase, each of which is a multiple subunit protein, and it is responsible for transcription of different RNAs. Coming to the holoenzyme, the holoenzyme is a complete and active enzyme having a polymerase with other cofactors. The holoenzyme of RNA polymerase in E. coli, it consists of 5 different units: alpha, that is alpha 2, beta, beta prime, omega, and sigma factors. The picture shows how the holoenzymes are present. Coming to this next slide, the table shows the RNA polymerase of E. coli. In E. coli, there are 4 subunits having different functions. The subunit alpha, the molecular weight in the Dalton is 36,512. The main function is to determine the DNA to be transcribed. The beta subunit, the molecular weight is around 1,50,618 Dalton. The function is to catalyze the polymerization. The beta prime, the size is around 1,55,613 Dalton. It binds an open DNA template. The another one, the final one is sigma. The size of the size or molecular weight is 70,263 Dalton. The main function is it recognizes the promoter for synthesis, start the synthesis or initiation of synthesis process. Coming to the recognition of the origins, especially in the prokaryotes, the each transcription region is called operon. One operon includes several structural genes like LAG-Z, Y, A, and upstream regulatory sequences. The promoter is the region, is the DNA sequence that the RNA polymerase can bind. It is the key point for transcription control in prokaryotes. Coming to the prokaryotic promoters, you can see that the left side is the 35 region of TTGA-CA sequence is the recognition site and the binding site of RNA polymerase. You can see the right side, that is the 10, nearly 10 region of the TOT AAT is the region at which the stable complex of DNA and RNA polymerase is formed. Coming to the transcription of the prokaryotes. What are the steps involved in transcription of the prokaryotes? There are three important steps or three important processes involved in the transcription of the prokaryotes. One is the initiation, elongation. Third one is the termination. What is the initiation? What happened in the initiation process? Here, the RNA polymerase recognize the promoter and start the transcription. So this place the transcription will start. Then second one is the elongation phase. Here, the RNA strand is continuously growing or elongated. The third process or step is called as termination. Here, the RNA polymerase to stop the synthesis of the nascent RNA. To stop the synthesis of RNA molecules, and the nascent RNA is separated to form the DNA template. So these are the three important steps involved in the transcription of the prokaryotes. Now we can see one by one. What is initiation? What happened in the initiation in the prokaryotes? The RNA polymerase recognize the recognition region that is it's contained the sequence called TTGA-CA, and it slides to that TAT, that is a prepromopause region or sequences, then the open the DNA duplex. The unwound region is about 17 plus or minus 1 base pairs. So the picture shows the RNA polymerase, how the unwind of the DNA in the double helical DNA structure. The first nucleotide on RNA transcript is always purine triphosphate. Normally here in prokaryote, GTP is more often than the ATP. So here the GTP structure remains on the RNA transcript until the RNA synthesis is completed. The three molecules form a transcription initiation complex. The following equations here how the RNA polymerase are working to form a transcription initiation complex. The picture shows how the binding and how the closed complex or how the open complex occur in the prokaryotic cells. And this picture also shows how the initiation or otherwise called as the transcription occur in the prokaryotic cells, and how the further the initiation may be start to the elongation form. Coming to the elongation, this is the second step of transcription in the prokaryote. The elongation, the release of the sigma subunit causes the conformational changes in the core enzyme. The core enzymes slides on the DNA template towards the third dash ending, because normally the transcription occur in 5' to 3' end. So here the free NTPs are added subsequently to the 3' hydroxyl of the nascent RNA. The below equation is so how the substrate NTP is added to the RNA strand to produce pyrophosphate or elaboration of the pyrophosphate ion. In elongation, the RNA polymerase, the DNA segment of nearly 40 nucleotide and the nascent RNA to form a complex called transcription bubble. The 3' segment of the nascent RNA hybrids with the DNA template at its 5 end it extends out the transcription bubble as the synthesis is processing. You can see the slide here. This picture shows the transcription bubble present in the process of the transcription. This picture shows the RNA polymerase of E. coli in the direction of the transcription or 5' to 3' direction of the transcription. Coming to the key points in the elongation. At any given time, about 18 base pairs of DNA are unwound, and the most recently synthesized RNA is still hydrogen bonded to the DNA molecule. It forms a short RNA and a DNA hybrid. What is that? It is form a short RNA and DNA hybrids. This hybrid is probably about 12 base pairs long, but it may be in shorter. The total length of the growing RNA bound to the enzyme and rDNA is about 25 nucleotides. This is the picture shows the simultaneous transcription and translation occur in the prokaryotes. The coming to the last steps in the transcription of the prokaryote is termination. That is termination of the synthesis of RNA molecule or terminate the synthesis of RNA molecule. Here, the RNA polymerase stop moving on the DNA template. So there is no movement of RNA polymerase in the DNA. It is normally stop the synthesis of mRNA molecule or the transcription process. The RNA transcript falls off from the transcription complex. The termination occur either row dependent or row independent manner at the end of the transcription unit. Now we can see what is row dependent mechanism and row independent mechanism in termination of the transcription process in prokaryotes. Here first we can see the row dependent transcription termination in the prokaryotes. This mechanism requires the presence of the row protein. So the row dependent mechanism, we need a row protein for termination process, and another case the row independent transcription termination, there is no need of row protein. Here now we can see the row dependent transcription termination in prokaryotes. This mechanism requires the presence of row protein, a RNA binding protein that form a hexameric complex at a particular signal sites, and it raises up the RNA strand. Once it reaches the polymerase, the protein complex displaces the polymerase from the DNA. That is RNA polymerase from the DNA. It terminates the transcription process. In this particular diagram, the system is shown as recurring hairpin to slow polymerase. Coming to the second one is sigma row independent termination, or otherwise called as intrinsic termination. Here, the termination signal is a stretch of 30 to 40 nucleotides on the RNA transcript. It consisting of many GC followed by a series of U, that is uracil. Next, the sequence specificity of this nascent RNA transcript will form particular stem loop structures. It terminate the transcription process in the prokaryote. So row independent transcription process, this mechanism does not require any row protein, but it requires two important elements. That is a hairpin formation, is a complementary section of the transcript RNA strand, followed immediately by a long stretch of T or A on the template. Coming to the transcription in the eukaryotes, The transcription in the eukaryote is similar to that in the prokaryotes. The process appear to be complex. Instead of one polymerase, RNA polymerase used in the prokaryotic transcription, here we can use 3 type of RNA polymerase for transcription in the eukaryotes. There are 3 RNA polymerase like RNA polymerase 1, 2, and 3 involved in eukaryotic transcription. So what is the role of RNA polymerase 1, 2, and 3? RNA polymerase 1 is localized to the nucleolus. It transcribes the RNA precursor molecule except 5S rRNA. RNA polymerase 2, most mRNA that encodes the protein and smaller nuclear RNAs. RNA polymerase 3 is involved the tRNAs, 5S RNAs, and other small RNAs. The mitochondria and the chloroplast have their own RNA polymerase. So coming to the slide, what are the differences in the transcription between the prokaryotes and eukaryotes? In prokaryote transcription, the polycystonic type of transcription is followed in prokaryote, whereas the monocystonic type of transcription followed in the eukaryotic genome cells. Normally, in eukaryote, the transcription occurs in the cytoplasm. In eukaryote, whereas in the eukaryote, transcriptions occur in the nucleus. In prokaryote, the transcription is coupled with the translation process. But in case of eukaryote, the transcription is occur in the nucleus and the translation is occur at the cytoplasm or is in the different places. Another difference, the single type of RNA polymerase required for synthesis of all type of RNA. Here in eukaryotes, the different type of RNA polymerase required for synthesis of all type of RNA. And another difference between the prokaryote and the eukaryote transcription, there is no need of any transcription factor for initiation of transcription in prokaryote. Whereas in eukaryote, the transcription factors play an important role, or it requires the transcription factor for initiation of the transcription in the eukaryotes. So in prokaryotes, the RNA polymerase are made up of 5 units, whereas eukaryotic transcription RNA polymerase are made by 10 to 15 subunits. So these are all the major differences in transcription between the prokaryotes and eukaryotes. So coming to the next part of this lecture is called as post-transcriptional modification, that is mRNA processing. The post-transcriptional modification is very important process in most of the eukaryotes because the pre-mRNA is normally processed or must processed, then only we can get the mature RNA through different steps or process is called capping, cleavage, addition of the poly A tail or methylation, etc. Now we can see how the post-transcriptional modification occur. What are the processes are involved in the post-transcriptional modification processing. In prokaryotes, already we discussed, most mRNAs have no modification. There is no special modification processing not needed in case of prokaryotes, but in eukaryotes, the post-transcriptional modification is one of the important process for getting the mature RNA. In eukaryotes, the synthesized transcript mRNA or heteronucleus RNA undergo the processing before leaving the nucleus. Before leaving the nucleus to the cytoplasm, formation of the protein or polypeptide, the process is called as translation. So here, what are the important processing undergoing the pre-mRNA to get a mature RNA? The first one is the addition of the 5' cap or capping. Second one is addition of 3' poly A tail. And the third one is splicing, methylation, etc. Now we can see the RNA processing. Very few RNA molecules are transcribed directly into the final mature RNA. Most newly described RNA molecule, that is called as primary transcript, is undergoes various alteration to yield the mature product, that is mature RNA. RNA processing is the collective term used to describe the molecular events allowing the primary transcript to become the mature RNA. Coming to the RNA processing at nucleus, what happened? The primary transcript, how convert into form a mature RNA is mainly due to removal of nucleotides. The second one is addition of nucleotides to the 5' or 3' ends or modification of the certain nucleotides. This picture shows how the transcription and adding and the process of 5' capping and how get the completion of the primary transcript and how the mature RNA get the process through the process called cleavaging, poly adding or splicing. First one is the removal of nucleotide by both endonuclease as well as exonuclease. Endonuclease to cut the specific sites within a precursor RNA. Exonuclease is nothing but just trim the end of the precursor RNA molecule. Is end of the precursor. So exonuclease act outside of the RNA molecule, just trim the ends. Endonuclease is cut the specific site within the precursor RNA molecule. So this general process is seen in the prokaryotes and the eukaryote for all type of RNA. The second one is the addition of the nucleotide to the 5' or 3' ends of the primary transcript or the cleavage products. You can see the picture, just add a cap, that is a 5' capping, poly A tile to prepare the mRNA. Third one is the modification of certain nucleotide on either the base or the sugar moiety. So add a methyl group to the 2' hydroxyl of ribose in mRNA and rRNA. It is extensive changes of the bases in transfer RNA or tRNA. Coming to the processing of mRNA in the eukaryotes. So we can see one by one, the eukaryotic pre-mRNA undergoes the extensive processing before it is ready to be translated at a cytoplasm. So the additional steps involved in the eukaryotic mRNA maturation create a molecule with a much longer half-life than a prokaryotic mRNA. Eukaryotic mRNAs last for several hours, whereas the typical E. coli mRNA lasts no more than 5 seconds. In eukaryotes, the mRNA is synthesized by the RNA polymerase II as longer precursors, that is a pre-mRNA. The population of different RNA polymerase II transcript are called heterogeneous nuclear RNA or otherwise called as hnRNA. Among the heterogeneous nuclear RNA, those processed to give the mature mRNAs are called pre-mRNAs. Pre-mRNA molecules are processed to mature mRNA by adding 5' capping or 3' cleavage and adding of poly A tile. Final one is splicing and methylation. This picture shows the overview of eukaryotic mRNA processing. Already we know that exons or introns are frequently are adjacently present in the genome. So you must remove the intron, then only the exon is exon or coding sequence may be start the transcription and translation to form a peptide or protein. So here you can see the first picture, the left side is exon, and in between the exon we have the introns. Okay, the end of the DNA molecule is called as the distal end is called as termination sites. So just adding up the 5' cap, primary the transcription we start, then go for the cleavage by the endonuclease, then adding of poly A tile with ATP, then the removal of the intron, the process called as RNA splicing, then you can get the mRNA. That's called as mature RNA. So these are the processes called as the pre-mRNA processing. These are the processes otherwise called as pre-mRNA processing. Coming to the 5' capping, 7-methyl guanosine, that is a methylated guanyl residues, is added to the 5' end of heterogeneous nuclear RNA via or through removal of the leading phosphate group at the 5' terminal by the RNA triphosphatase. Then transfer of the guanosine monophosphate, that is a GMP, from the guanosine triphosphate group by the guanylate transferase. The methylation of the guanine by the guanine 7-methyl transferase, that is a methyl group from the S-adenosyl methionine, are simply called as SAM. What are the function of the cap? We must know what are the function of the cap. The cap provides the protection from the some ribonucleases. It enhances the translation. It enhances the transport from the nucleus. It enhances the splicing of the first intron for some mRNAs. Finally, it prevents the exogenous degradation. These are the some of the functioning of the cap. Next is the second one is the 3' cleavage and polyadenylation. In most pre-mRNA, the mature 3' end of the molecule is generated by a cleavage followed by a addition of a run, that is addition of a run or tile, that is the A residues, which is called as poly A tile or addition of the poly A compound. RNA polymerase 2 does not usually terminate at the distinct site. Next, the 50 to 250 adenyl residues, that is the AMP, are added to the 3' end of the heterogeneous nuclear RNA through cleaving about 20 nucleotide downstream from an AAU-AA recognition sequences. Then the addition, that is the extension up to 250 nucleotides of poly A tile, that is generated from the ATP by the poly A polymerase. This is the picture shows how the addition of the poly A tile, how the transcription may be started by using RNA polymerase. Coming to the function, it is normally prevent the degradation in the cytosol by using exoribonuclease. It stabilizes the mRNA molecule. This picture shows the post-transcriptional modification of the RNA, just 5' capping and adding of poly A tile at a 3' end. This prevents the degradation of the mRNA in the cytosol. So then the pre-mRNA splicing and methylation. Eukaryotic genes are composed of exon, which corresponds to the protein coding sequence, or otherwise called as this coding sequence is a code for protein or amino acids. And another part in the eukaryotic is called intron, which may be involved in the gene regulation but are removed from the pre-mRNA during the processing. What is methylation? A common modification that alters the life cycle of transcripts. What are the key points of the post-transcriptional modification is very important. We must know one by one. The first one is the 7-methyl guanosine cap is added to the 5-N. That is the 5 is otherwise called as 5' capping of the pre-mRNA while elongation is still in progress. The 5' gap, the 5' gap or 5' capping protects the nascent, that is the newly formed RNA or mRNA from the degradation and assist in ribosome binding during translation. It's assisting in ribosome binding during translation. Okay, third one is the A poly tile is added to the 3'N of the pre-mRNA once elongation is completed. So in the proximal end, the 5', the 5' capping is normally followed. The 3'N, just we add poly A tile. The poly A tile protects the mRNA from the degradation. It is very, very important one. You must remember. It aids in the export of mature mRNA to the cytoplasm and is involved in binding the proteins involving in initiating the translation process. Normally, introns are removed from the pre-mRNA before the mRNA is exported to the cytoplasm. So, in this lecture, we covered what is the transcription in prokaryote and eukaryotes, what are the steps involved in the prokaryotic transcription. There are three important steps. One is the initiation, elongation, and termination. What are the difference between the transcription in prokaryotes and transcription in eukaryotes? Then the next part of the lecture, we discussed about the post-transcriptional modification. Normally in prokaryote, there is no need of the post-transcriptional modification in mRNA in prokaryotes, but in case of eukaryotic, the post-transcriptional processing is very important. There are different ways the post-transcriptional processing is involved, and to make a pre-mRNA to the mature RNA. Normally here some of the process like 5' capping, then addition of the poly A tile, and then splicing of splicing or removal of the introns in the eukaryotic genomes, then methylation. These are some process is helpful to process the pre-mRNA to the mature RNA, and then the mRNA released to the cytoplasm for translation. Thank you, one and all present here.